Hey everyone, welcome back to the second part of our conversation about neurons and neurotransmitters. So, hopefully, by now you've watched the lecture about kind of what a neuron is, what that cell looks like, some of the different structures of a neuron, and how that operates. So, if so, this next slide is kind of a review for you. This might not be as beautiful as the drawing that I drew for you, but you can see these different parts of the neuron and hopefully your notes look something like this. So, quick little review. We have the message that travels away from the soma or the cell body down our axon. The axon is encased in this fatty substance called the myelin sheath. When it gets to the end of the axon, it hits what's called our axon terminals or terminal buttons. That electric message transitions into a chemical message, releases chemicals into our synapse. It's that gap in between two neurons, and then they bind with the dendrites of a neighboring neuron, what we call the postsynaptic neuron. That's all from our first lecture. So let's go into a little bit of detail now on the different types of neurons that we have in our body. So neurons are found in our nervous system, and there are three main types that you need to know for this class. The first type, I'm actually going to skip a little bit ahead. The first type is what we call an interneuron, and the interneurons make up our brain. Our brain is made up solely of interneurons, and what the interneurons do is kind of two things. They bridge the connection between sensory and motor neurons, and they also allow us to process information. So let's take, for example, we have this graphic on the screen of someone touching a candle. First of all, that's a bad life decision, but for whatever reason, a person has touched a candle, and that flame of the candle is going to cause a message to be sent from the tip of your finger up towards your brain, and we call those sensory neurons. We have sensory neurons all over our body. They're all over our skin, they're in our eyes, in our nose, our mouth, in our ears. And our sensory neurons are responsible for sending messages toward our brain. So that message travels up your hand, up your arm, toward your spinal cord through what we call sensory neurons. Motor neurons kind of do the opposite, though. Motor neurons are sending messages from our brain to our body. So when you withdraw your hand back from that candle flame, your ability to move your hand, think MO, move your hand, those are motor neurons. Just like the motor can help a car move, your motor neurons help your body move. Now, I have in parentheses afferent and efferent. Those are just some fancy words for the exact same thing. So you might see sensory neurons written as afferent neurons with an A, and you may see motor neurons written as efferent neurons with an E. Easy way to remember this, think they are the same, S-A-M-E. Sensory is the same as efferent, motor is the same as efferent. And then again with the interneurons. Interneurons are found in our brain and in our spinal cord. And something that's kind of cool, we have these interneurons in our spinal cord, right? So for reflexes, like when you touch something hot or if you step on something sharp, the way that reflexes work, like a true physiological reflex, you can have a message travel through your sensory neurons toward your brain and spinal cord. And when it hits the interneurons in your spinal cord, a message will be sent down toward your hand or your foot through motor neurons without even needing cognition, meaning you can withdraw your hand from that hot candle flame even without your brain telling you to do it. That's why it happens so incredibly fast, because the interneuron will turn it around and send a message through motor neurons to your hand, your foot, or whatever it may be. And so we call it physiological reflex. Now, how do we send the actual message, right? How does this electric message travel down a neuron? Well, this is not an AP biology class, but we do go into some kind of detail specifically looking at these cells. So there's some things that you need to keep in mind. First of all, remember neurons are cells, and when they're at rest, they have a negative charge. And the reason that these neurons have a negative charge is because along the inside of the neurons is primarily negative potassium ions. So we have potassium ions that are inside of our cell. And then surrounding the neuron, we have positively charged sodium ions. So I'll say that again. Sodium ions are located on the outside. Potassium ions are located on the inside. Think a salty banana, right? If you've got a salty banana, you're going to have sodium on the outside, the potassium on the inside. And that's when a neuron is at rest. What do I mean at rest? I mean, it's not doing anything. Like you've got neurons on your shoulder, right? On the skin of your shoulder. Right now it's not doing anything, but if someone comes and taps you on the shoulder, it excites those neurons and all of a sudden they're moving, right? They're acting. So what happens? Well, what happens when we want to excite a neuron is these neurotransmitters will travel across the synapse. They'll bind with a neighboring postsynaptic neuron and get to what we call a threshold. Think of it almost like a minimum amount of stimulation that is required for that next neuron to get excited. If there's a few neurotransmitters, it's not going to do anything. A few more, it's not going to do anything. But at some point, it's going to fire. Think about a toilet, right? You're going to slowly push down on the plunger to flush the toilet, and then at some point, boom, it flushes, right? Think about firing a gun. If someone is slowly pulling a trigger, at some point, the gun fires, right? It's reached the threshold and it's fired. That's the exact same way that a neuron works. Neurons work on what we call an all or none response. A neuron either fires or it doesn't. So you have these excitatory neurotransmitters that are binding at the receptors, and at some point there will be enough of those neurotransmitters that it hits the threshold, the minimum amount, and then boom, that neuron will fire. When the neuron fires, there's something interesting that happens with those ions. We got the negative ions on the inside, we have positive ions on the outside. Well, that axon wall is what we call, it's selectively permeable as far as what is allowed in and out of that axon wall. So what happens when a neuron fires is those positively charged sodium ions are going to flood inside of an axon. And temporarily, the neuron is going to be what we call depolarized. Think about what the word polar means. Polar means separate or opposite, right? North pole, south pole. When a neuron's at rest, all those ions are polarized. A neuron at rest is polarized. You've got positive sodium ions on the outside and negative potassium ions on the inside. When a neuron fires, it becomes depolarized, meaning those ions come together, right? And that process of them coming together, this depolarization process, is what we call an action potential. It's the action of the neuron. So we've talked about when a neuron is at rest. We've talked about what happens when the neuron fires, when there's action. And then the only other thing here is it has to reset, right? Like those sodium ions that are now on the inside of an axon, they need to get pumped back out. They get pumped through what we call these sodium-potassium pumps. And so the sodium ions need to get pushed back outside of the axon. And we call that the refractory period. It's like hitting the reset button or reloading a gun, right? If you fire an old musket that just has one little musket ball in there, well, you got to reload before you can fire it again. It's the same way with our neurons, right? Back to the toilet example. If you flush a toilet, you got to let the back of the toilet bowl fill up again before you flush again. So this reloading is what we call the refractory period, and it is allowing the neuron to go back to at rest and wait to be fired again. And keep in mind how quickly this happens. I mean, it happens in a fraction of a second. Pretty amazing. So a little bit of a reminder, I talked about this in our last lecture as well. As the message travels down the axon, when it gets to the end of the axon, this is just kind of zoomed in on what's called the axon terminal. And we have these little vesicles, these little bubbles here, right? And the vesicles get pushed up against the axon's wall here, the terminal wall. The neurotransmitters are released into the synapse, and then they bind with the receptors of the neighboring dendrite. Remember, neurotransmitters, those are the chemical messengers, and we're going to talk about some specific ones here in just a second. They work like a lock and key, a very specific pattern. And any neurotransmitters that are not being used, they get recycled back up through reuptake. So that's review from our earlier lecture. So let's actually take a look at those chemicals themselves. You have dozens and dozens of different types of neurotransmitters in your brain. These are, the 6 or 7 that you got to know for AP Psych. I'll go through some of this. I'm not going to go through every single detail because you can kind of read this on your own, but some things to keep in mind. Acetylcholine, incredibly popular neurotransmitter that the AP exam likes to ask. Acetylcholine really does two big things. Acetylcholine helps with muscle memory, and acetylcholine helps us, with muscle action, move your ability to walk across the floor, right? Your ability to throw a baseball, that's all acetylcholine. And also learning things and with memory. Muscle contraction, when your muscles contract, that's because acetylcholine is being released in synapses. And if you don't have enough acetylcholine, the result can be Alzheimer's disease. Dopamine. Dopamine is released in your brain to essentially reward you. Dopamine makes people feel good. And so it's associated with things like learning, because if we eat a cheeseburger when we're really hungry, our brain releases a bunch of dopamine. If I'm really, really thirsty and I drink some ice water, my brain releases dopamine. It says, do that again. So dopamine is associated with our ability to learn. If we have too much of this dopamine, however, it can lead to hallucinations, and there's associations between an excess of dopamine and experiencing schizophrenia. Also recognize that dopamine is highly associated with addictions. There are lots of different drugs that work by releasing dopamine in the brain. Dopamine makes you feel good, and so people can become highly addicted because of the way that their brain is making them feel with this excess of dopamine. Serotonin, for AP Psych, you want to tie together serotonin with depression. 99% of the time, if you are seeing a question in this class about serotonin, the question's probably going to be asking about depression, specifically that you don't have enough serotonin. So we found that a lack of serotonin often leads to people experiencing depression. Serotonin really helps with our ability to control our moods. Norepinephrine and epinephrine, for the sake of this class, you do not need to know the difference between those two terms. They are structurally different. If you look at the chemical makeup of them, they're different. But for this class, you will see them being used interchangeably. So norepinephrine, epinephrine, what does it do? It puts our body into high alert. They are what we call excitatory neurotransmitters, and it puts the body into a heightened state of arousal. And what I mean by a heightened state of arousal is, we increased pupils, or where we have our dilated pupils, increased heart rate, deep breathing. It's preparing our body for action. Maybe we need to flee from something dangerous. Maybe we need to fight something dangerous. That's what we do with norepinephrine. Gamma aminobutyric acid, or what we call GABA, is the major inhibitor neurotransmitter. Slows the body down. And I always remind students, think GABA as get a break adjustment. If you are anxious, right, and your heart's beating really fast, well, you want to kind of pump the brakes a little bit. You want to slow the body down, and GABA allows us to do that. So it's no surprise that if someone, for example, has too little GABA, that they may be more likely to suffer from OCD or from some anxiety disorders and things like that. Endorphins is like the natural Tylenol in our brain. Endorphins help us with pain control. And so if you stub your toe in the morning getting dressed, your brain is going to get flooded with endorphins to help you control that pain. And if you have too much endorphins, then you can experience these kind of artificial highs. You might be familiar with a runner's high, for example, with distance runners who run 12, 13 miles at a time, and they get to a point where their brain is releasing so many endorphins that it actually overcomes the experience of pain, and they actually start to feel pretty good. And then the last one up there is the most popular neurotransmitter in the brain, the most abundant neurotransmitter in the brain, and it's what we call glutamate. It is a major excitatory neurotransmitter. It is highly involved with memory. Know that if you have too much glutamate in the brain, it can lead to things like seizures. You'll see that acetylcholine and glutamate look pretty similar. It's unlikely you'll have to differentiate those two. Look for perhaps those other facts that helps separate them, or if we're talking again like muscle movement, your ability to do something physical with your muscles, walk around, swing dance, whatever, it's probably going to be asking about acetylcholine. So went real fast through that list. Don't neglect your reading and kind of going into some more detail about these neurotransmitters, but hopefully this helps get you started. If you have any questions at all, don't hesitate to ask. And as always, good luck.